Okay, so that's, getting that out of the way. So now we know, okay, we have high dimensional data and we have redundancy in the data that comes from, measuring the same thing kind of in like redundant ways. So the dimensionality reduction is really simple, that we want to take high dimensional data and then somehow transform that original data into something that's simpler and has fewer dimensions. And as we said, the goal would be to make it more explorable for like humans. And as I said before, machine learning algorithms can deal with high dimensions fairly well. So that's not like a big worry, but they can be potentially overfitting. So that's nice. And then, so what we talk about specifically in the next, in a moment, is principal components analysis. So this is like a really popular method that's completely rooted in linear algebra and will not really go into the math of it, but it has a nice way of dealing with redundancy between variables that's linear in nature. So as I said, PCA, principal components analysis, is a technique that can be used for dimensionality reduction. It isn't necessarily a dimensionality reduction method in itself. It basically just changes the dimensions of your data, and it kind of rotates your data in high dimensional space. And then it allows you to cut away dimensions that seem less important in your data. And we'll show that in a two-dimensional example in a second. And so that has a lot of applications. So for example, if you have noise in your data matrix, you can calculate PCA and then you just remove part of the dimensions of your new dimensions that explain a very low amount of variability in your original data. And those are potentially the principal components that are introduced by noise in your measurements. So here's a really simple example. So this is obviously not high dimensional. It's a two-dimensional data set. And it has fairly high redundancy between the two dimensions x and y. So what principal components analysis does is it kind of finds the axes, like it will in the first step find the axis that explains the most amount of variance. And as a human, when you look at this, you immediately fit a straight line through these points in your head, right? And so that line that you're kind of drawing with your own, like, imagination, that's kind of the first principal component. It's what it's called. And this axis, basically describes most of the variance in this two-dimensional data set. So if we would just use this information, the first principal component, we could basically and remove the second dimension, the second principal component, we would maintain most of the variance from our original data. So how this looks is we have the first principal component, we find the second principal component, and these principal components are the new dimensions, if you want, of our data. And they have this property that they have no shared information between them, right? So they're orthogonal to each other. And that's exactly what we wanted, right? So in that way, if we now, remove principal component 2, the first principal component maintains most of the information from the system. So in practice, how this would look like is we rotate our cloud in this high dimensional space into these new dimensions, which were principal component 1 and principal component 2. So now PC1 is basically X star and PC2 would be Y star. And so now we could technically just flatten the data down. So if we just remove the Y coordinate, we end up with a one-dimensional data point. but we spread out as much information as possible on that dimension. And there's some examples in code. So like programmatically, this is super easy and very efficient to compute. So this is this used to be the most popular method before like some others were invented. The nice thing about these transformations is that the space in between these data points, you can really interpret because it's kind of a combination of your original dimensions. So that has like practical applications. So this used to be like the gold standard of doing dimensionality reduction and showing high dimensional data in like a two-dimensional way, if you want. Okay, so now I kind of show you how principal components analysis works in Python. And for that, we basically first have to have some kind of data set. So we create a synthetic data set that is high dimensional. So in this example, it will have 100 dimensions. And then we apply PCA, and we want to extract the top 2 non-redundant dimensions. And then we see how well we can retrieve the original signal from the data. So really simple, on the left, you see the data that we generate. It's basically just 3 multi-normal distributions in 2D space. And then each cluster has 50 data points. And so this is our original data. And now from this data, we create a new high dimensional data set. And so how we do this, I'll just really briefly go over it. So here we have a for loop, we iterate over it 100 times, and we build linear combinations from the original 2 dimensions, right? So initially we had 2 dimensions from our nice clusters. And now what we do here is we multiply a random normal float number to the first dimension and then add the second dimension that's also multiplied with some random number. And so what this does is it creates a new dimension that's a linear combination from our original data. And then we also add like a good amount of noise for good measure, right? And then so we do this 100 times, so we get 100 dimensions that are somewhat based on the original 2 dimensions that we had. So if we do this and we just plot 2 dimensions, like 2 random dimensions that we just created, it looks somewhat like this. So it's kind of messy, right? So you can't really see that the yellow cluster and green cluster, they kind of overlap. So it wouldn't be that easy to really pull them apart. So that's where the high dimensions are somewhat problematic. So like means basically we can't just take 2 dimensions and look at it and we figure out what's going on. So now if we run principal components analysis, it's really easy. So you have to import obviously the packages, but basically what you do is here you initialize this PCA object, and here we tell it to give us the top 2 principal components, meaning those are the principal components that have the most variability. And then we transform our new data into this reduced space. So we, as we showed before, we rotate the space around, and then we remove the other dimensions, so like the other principal components. We don't want those. And then once we have this is our reduced data. So now we can plot that, right? So here we have, in this plot, we have our original data, right? This was before we created the new 100 dimensions. And then in the second plot, we have our reduced data that we got from calculating the first 2 principal components. And so what you can see is that the algorithm was, really good at getting back the original clustering. Okay, so that's kind of what this looks like synthetically, and this is kind of also how it works in practice when you work with, gene expression data, for example. So And then this plot is how it looks if we look at the explained variance for the dimensions of our fake data, right? So we had these 100 synthetic dimensions. So this plot shows us the percentage of variance that's explained by each principal component, ordered from the most important to least important. And so in this example, the first principal component explains more than 60% of the variance in the data. And the second one, it's somewhere here, right? So it's maybe 15% of the variance. And then all the other dimensions, they're basically completely unimportant. So the algorithm was really able to figure out what the true original dimensions were. And that's why the reconstruction worked so well. Okay. So now we're talking really about this. So you get a feeling of what dimensionality, the intrinsic dimensionality of data is, right? So imagine you have a random data, it's completely random. And we have 1,000 by 1,000 values. So we have 1,000 dimensions and 1,000 data points. So if we show exactly the same plot of these principal components, right? So they're ordered from left to right. And we plot the explained variance of each of the principal components, you see that on completely random data, this is the distribution that we get, right? So this is just on random data. That's what we would expect randomly, right? So just so you get an idea. So it's not like uniform or anything like that. It's just, decreasing pretty quickly. So now we can also create data points, kind of similar to before, where there's some redundancy between dimensions. And so if we do that, and then we calculate the principal components, we see that the first principal components get more weight compared to the other principal components, right? So the algorithm is able to identify this redundancy and then shift the information into these, first principal components. And then we can go even further if we had like a good amount of redundancy, it gets even more significant. And then you see this this small kink here is because when I created this data, there were only 500 truly independent dimensions. And then I did the same where I built these linear combinations of those dimensions. So the algorithm at 500, all the other principal components have no variance anymore, right? They're just not important at all. And so now if I do high redundancy, so in this case, I had like 200 dimensions to start off with, and then I build these 1,000 dimensions from those. Then the principal components, the variance explains falls even faster. Okay, so this is synthetic data, so that's great and all, but like how does that actually translate to the data that we work with? Right, so just to recap, right, we built this synthetic data, and then we created through linear combinations new dimensions, and then we kind of started with 1,000 original dimensions, 500 and 200. So that's where the redundancy comes from. And then, so ArchES4 is a resource that Avi mentioned before that kind of collects all publicly available RNA-seq data. And so right now we have about 500,000 human and 500,000 mouse RNA-seq samples. It's pretty comprehensive. And each sample, so each data point, has 60,000 genes about, right? And so you can think of the genes as the dimensions of the data. So the question would now be is what is the intrinsic dimensionality of the data, right? So it's clearly not 60,000 dimensions, right? So we can probably compress the data quite a lot and still maintain most of the information, right? So that's the question, right? So how does gene expression compare to synthetic data? So if we look at this plot, right, where do you think actual gene expression would fall in, right? So it's slightly different, since gene expression in this case has 60,000 genes, and here we only have 1,000 dimensions to start off with. But so does anybody have an idea what like where this plot would fall for actual gene expression? Anybody? I'll pick on somebody. Okay, let's pick on let's see, Meredith.
I would guess somewhere in between medium to high redundancy. I would assume that, like, if you're looking at a set of cells, most of them are going to be expressing similar levels of like most of the genes.
Yeah, so the example I will show is basically a cross-section of any type of cell. It's like completely randomized data set, right? So there could be more variability than if you just look at like a tissue type, right? Okay, so maybe Jonathan, what do you think?
I think there would be a good amount of redundancy.
That's for sure, yeah. So do you think it's also like between medium and high, or?
I'm gonna go towards high.
Towards high, okay. And then let's get one more for good measure. Maria, what do you think?
Yeah, I think I agree. Also, somewhere between medium to high redundancy.
Okay, so yeah, so it's right. I mean, maybe people have already seen that genes correlate quite a bit, right? So the actual redundancy is very high. It's way higher than the data that you've seen before, right? So the truth is that you can basically explain gene expression with very few dimensions. Like, so you, like, if you reduce your data down to 10 dimensions, you capture way more than 95% of the variability in the data. So that's really, I think, an important insight. So you can kind of like remember that, right? So even though we have these, 60,000 genes, they really move together in like big groups, right? So you have a gene and it will be correlated to 1,000 other genes. And so the real dimensionality of this data is quite low. And it kind of makes sense, because if you think about it, like how many cell states are there really, right? So cells usually, as a energetic system, they kind of find this global or like local minimum that they fall in, and that's the cell type, right? Or like basically the cell type, and it kind of maintains that equilibrium, right? So like a lung cell will stay a lung cell, right? Or like a skin cell is a skin cell. So, and then there's of course mechanisms underlying these cell types that are variable to a certain degree, but in the end there is like a finite amount of states that a cell is really in, and then the rest is like a lot of noise. But even if you have, let's say, 20 dimensions, there's a huge amount of information that you can store in 20 dimensions, right? So that's why you don't really need that much to explain gene expression and to really explain a cell type. So this is from a nice paper. It's called A Global Map of Human Gene Expression. And it looks at this specific hematopoietic system. And what's really nice about this is, so they're interested in kind of how malignant the cells are, and it's kind of perfect, because on the first principal component, which is the x-axis, it really splits between like the different tissue types. So we have these different clusters. So you have, for example, the connective tissue on the right, and on the second principal component, it really stratifies over the malignancy of the cells. So this is a really cool example where you have this high dimensional data, and you just take the first 2 dimensions, and it explains basically the story that you want for like a nice publication, right? So I'm sure this was yeah, in Nature Biotechnology. So it's like a good journal, right? And it's like really illustrative, like how powerful these methods are, right? So you can see everything in 2 dimensions. I mean, not everything, but like, you know, it makes it really visually striking. And so in reality, this is exactly how this looks like, right? So we have these data points. They're basically a very high dimensional data set, right? So we have the samples, which are the points. So the rows in this matrix are the points in these plots. And then in this case, let's say there were like 17,000 genes that were measured. And so these 17,000 genes can be compressed into 2 dimensions. And we kind of see everything that we want. So this is also like a very nice visualization of dimensionality of data. So this is a data set from the Cancer Genome Atlas. And for this project, they study a lot of different cancers in humans. And in this case, this is the breast cancer data set, where they have 1,100 samples. And they measure not only gene expression, but also methylation and protein levels and all sorts of other ways, like methylation. So the breast cancer samples, you can differentiate them into different subtypes. So there's luminal A, luminal B, triple negative, and HER2 positive. And so these are usually identified by different markers. So this is the table that kind of explains how they're defined. And so if we look at the actual dimensionality within these different data sets, we can see that they have a different intrinsic dimensionality. So the luminal A subset in RNA-seq, for example, has kind of the lowest intrinsic dimensionality. So the plot really briefly explain what it does is, in the case of luminal A, there were about 400 samples in RNA-seq that were available. And if we take those 400 samples, and then we ask how many dimensions do we need to capture, let's say 98% of the variance, how many dimensions do we need? And then it was somewhere between 40 and 50, right? And because we only have a finite amount of these samples, a way to kind of predict the, the total dimensionality of this kind of subtype, we can kind of interpolate it by subsampling those 400 and then doing this, like asking this question over and over again. So we can kind of extrapolate how many dimensions we would need if we had an infinite amount of luminal A samples. So it's a kind of fun way to do that. And so you can see that luminal A had like the lowest predicted dimensionality, and then subtypes like HER2 positive, they seem to have the highest dimensionality. And so this is also similar in the different data types that we have. So in methylation, it kind of has the same order, even though it's less clear because there's just less data, so it's harder to extrapolate out. But so now you basically have a rough idea what these dimensions look like and what's like intrinsic dimensionality is.